All right, so good evening, everyone. Welcome to another of our Café Scientifique presentations. My name is John Willis, and I will be your host this evening. Before I introduce this evening's speaker, let me give you, as usual, a couple of information messages. So here we are in June. This is our third month of online Café Sci. In the last month, recent innovation is you can now join us on YouTube as well. So once we actually give these talks, we record them, and then typically the day after, we post them on YouTube. And you can search for us at UVic Café Sci, and that will get you to our channel. And that's a great way to also to recommend us to those friends and colleagues of yours who aren't able to join us on the evening, but still want to engage in these talks. The other piece of news is that Olav, my co-host, and I have decided we're actually going to take a break in July and August. We're going to probably, like you guys as well, go off on our BC vacations. But that's also going to give us the chance to put together a great schedule for you starting in September. We fully anticipate, no surprises, that we're going to be continuing with online presentations. We don't see us returning to Herman's Jazz Bar anytime soon, unfortunately. But I think over the last 3 months, we've demonstrated that we can give some great talks online and keep a great sense of community. Which leads me to this evening's presentation. So our presenter this evening is Dave Leitch, a new professor at UVic. You joined us in 2019, is that right, Dave?
That's right, yeah, the beginning of 2019.
Now, I just have to, while I'm giving this brief introduction, I saw a great way to introduce you on your own group website at UVic, and you describe your group by saying, "We in the Leitch group are chemical cartographers, striving to map and understand chemical reaction space in the realm of organic synthesis and catalysis." How could you possibly give a bad talk on the back of that kind of setup, eh? You're a true chemical explorer. And so the title of Dave's talk this evening is Developing New Medicines for COVID-19. Are we there yet? So Dave, over to you.
Great, thank you, John. So tonight I am not going to talk at all about my group's research or really a lot of my kind of primary research interests at UVic right now. Rather, what I'd like to do is give a contemporary, cutting edge, up to the last minute overview of basically where we are with respect to developing new medicines for COVID-19. So just in terms of a little bit of context as to why a self-proclaimed chemical cartographer would be giving this kind of a talk. Prior to coming to UVic, I worked in the pharmaceutical industry in the United States for about 5 years. So my background in that was in drug development and sort of more on the manufacturing side of things. And so I have a chemist's perspective on the process of medicines development, which again is what I'm going to talk to you all tonight about. Just a disclaimer, I am clearly not a medical doctor. I am not an epidemiologist. I am not an infectious disease expert, even though the ranks of these, especially on social media, seem to have swelled a lot in the last 6 months or so. So again, this is going to be coming from a chemist's perspective. So 2020 so far, if it's feels anything to you like it does to me, we seem to be living through about 5 consequential years in 20th century history, all slammed into about 6 months, at the beginning of the year here. The Chinese authorities informed the WHO of the existence of what we now call COVID-19 on New Year's Eve 2019. So we are 6 months down the road here, and unfortunately, the disease is very much still with us, and it's still a huge worldwide problem. There is a lot of hope being placed right now in developing new medicines to help combat this, basically culminating in hopefully in some kind of a vaccine. There are a lot of potential options here, and so these are the things that I'd like to talk to you about this evening, and also again give a very up to the minute update of where we are at the moment. So it's a bit of kind of interesting historical coincidence that almost exactly 100 years ago the world went through something similar. The so-called Spanish influenza at the end of World War I swept across the globe ultimately resulted in 50 to 100 million deaths or so we estimate now. This is clearly one of the most consequential events in human history, and certainly in 20th century history. And while the fact that in 1918 and 1919 we knew that what we were facing was influenza, we now know a lot more about this particular disease than they did at that time, obviously. So we now know that the influenza is caused by a virus, and that the particular strain of virus that was so virulent at this time was the H1N1 variant. Now, again, in 1918, 1919, the progress of medical science was still, modern medical science was still in its infancy. While the existence of viruses was recognized in the early 20th century, several had been studied and even isolated prior to this time. The human influenza virus wasn't actually isolated until 1933. And so in terms of actually knowing what was, what needed to be dealt with and possible treatments for it, there really weren't a whole lot of options. Most of the things that we would use today to combat something like a flu pandemic, flu vaccines, antivirals, antibiotics to treat things like secondary infections, these just were not available at the time. And so we can look back at how the world dealt with this 100 years ago, but obviously now, flash forward 100 years to today, we have very sophisticated medical science. We have fully developed fields of virology, pharmacology, epidemiology. We've, in the intervening time, we've eradicated smallpox. We've nearly eradicated polio. We've subdued influenza that caused so much pain and anguish 100 years ago, such that the second H1N1 pandemic about 10 years ago in 2009 actually turned out to be no worse than a typical flu season. Again, because of all of these advancements that we've had over the last 100 years. We've turned HIV into basically from a death sentence into a manageable chronic condition. We've cured hepatitis C. And while we haven't cured cancer yet, we have made amazing progress in treating, diagnosing, and ultimately giving, a lot of hope and a lot of life back to folks who had at one time would have been considered, a sure terminal diagnosis. So in the year 2020, in the face of a new, deadly, highly infectious disease, based on all of the miraculous and important discoveries that we've made over the last 100 years, where are we in terms of developing weapons against this new virus? Even though it's only been 6 months, it feels, again, at least to me, like we've been on this trip forever. Are we there yet? Do we have options that we could start to deploy to medically treat or prevent this disease? So for whatever reason, humans like to frame our struggles against, our difficult struggles as in terms of a war. We have, certainly I've seen many headlines about the war on COVID-19, the war on SARS-CoV-2. And so if we're going to frame this in terms of the war metaphor, we might as well consult, the expert Sun Tzu and The Art of War. A former colleague of mine at GSK, the pharmaceutical company that I used to work for, he would love to misquote Sun Tzu pretty much once a day. And so I have this very close association between pithy quotes from the Art of War and pharmaceutical development. And so a particular quote that we can creatively interpret here to apply to the current situation is that effectively Sun Tzu is saying that we need to know our enemy, and we also need to know ourselves if we're going to be successful in this battle. Now, knowing our enemy in the current context is relatively straightforward. We know that there is a novel coronavirus that is causing the disease that we call COVID-19. And so in terms of the specific details about knowing our enemy. Understanding all that we can about this virus, what its structure is, what its functions are, how it infects the cells in our bodies, how it's transmitted. These are all the important things that we need to know before we can hope to be successful. And as we'll see in a second here, this is actually the easy part. A lot of this work has already been done, and it has been done incredibly quickly, and we know exactly what we were dealing with. Knowing ourselves in this context is it takes a little bit more interpretation, and notably, at least in this particular translation of this quote, there's nothing about knowing our enemy but not knowing ourselves. And so I kind of take this to refer to being confident in our scientific methods and being confident in the information that we're generating about these potential treatments. And here again, even though Sun Tzu doesn't have an outcome if you don't know yourself but know your enemy, Richard Feynman, who was a very famous physicist, Nobel Prize winning physicist and kind of quirky popular author, has a quote that kind of fits in with this theme that is really, really important just in science in general, but specifically with respect to developing new medicines. And that's that we cannot fool ourselves, even though it is really, really easy and really tempting to do this. And so we will see examples of how that can be a real problem, and how we need to be confident and be true to ourselves and be sure of ourselves when we are doing something like developing a new medicine. So let's take a look at our enemy, the SARS-CoV-2 virus. Again, this is the virus that we know causes the disease COVID-19. Viruses, just in general, are extremely strange things. By all reasonable definitions, they aren't actually even alive. In order to be alive, something has to be able to self-replicate, and viruses are not capable of doing that. They infect our cells in order to make copies of themselves. Really, you read a kind of dispassionate description of what a virus is, and it sounds more like something out of a science fiction novel. A pathogenic nanomachine that hijacks living cells to produce more copies of itself. So viruses, again, they're quite simple. They're not capable of reproducing. But let's take a look inside here and see what we could possibly use as a weak point, or some piece that we can leverage to target our potential therapies to. And so the SARS-CoV-2 virus is basically just a capsule made up of protein and other stuff that encloses its genetic material in the form of RNA. RNA and DNA, these are the blueprints that life uses to describe how to create a new organism, a new cell. And so this particular virus, again, has its RNA genetic material enclosed in this capsule, and that RNA tells the host cell, the cell that's been infected, how to produce the 29 individual unique proteins that are the structural and functional pieces of this virus. So again, if we follow with this sort of blueprint analogy, if the RNA here is the blueprint, then the proteins are everything else about the building, everything from the steel girders to the elevators to the doors to the maintenance staff, security system, et cetera, et cetera. And so these proteins are what we are going to target as weak points in terms of developing new medicines. And so the more that we know about the virus and about these proteins, the easier it will be, and the more confident we're going to be able to be with respect to creating a new treatment. So if we can either disrupt the function of one of these proteins or several of these proteins, or attach something to one of the proteins that will tell our immune system to attack this and wipe it out, or even to just attach something to block a protein from doing a particular function, then we can stop the virus from infecting more cells and ultimately cure the disease. Now, one of these proteins, probably the most famous protein in the world at the moment, is this so-called spike protein. So it's quite prominent. It's out on the surface. It's the red protrusions on the images here. And so this is the part of the virus that allows it to enter into human cells and dump its blueprints into that cell to hijack it for production. And so if we could block that spike protein somehow, if we can disrupt it, if we can stick a cap on it, or something like that, then presumably we should be able to stop the virus from infecting more cells. Another important set of proteins, and they're not shown explicitly on here just for simplicity, are proteins that will actually duplicate the virus's RNA. So it's just a copying machine for the blueprints that are inside the virus. This is another potential weak point that we could exploit. If we can find some way to kind of toss a molecular wrench into that copying process and disrupt it, then the virus won't be able to replicate, and then it will die out, and we won't need to, the infection will be stopped. So all of this information that we have about this virus has come within, we only knew about this thing about 6 months ago, starting 6 months ago. All the information that we have, so we've been able to isolate, map the genetic code, structurally, get a look at the different proteins. So the spike protein in particular, we know the molecular structure of this down to pretty fine detail. We've mapped all of these different proteins, the 29 proteins again that I mentioned that are part of this virus. We've actually been able to map how those proteins might interact with human proteins. We have electron microscopy images of this thing. Again, within a really, really short period of time, we know exactly what we're dealing with. And this is one of the testaments to where we are in 2020 with respect to medical science and biological sciences. That this did not exist on our radar at all 6 months ago, and now we know a ton of information about it that we can hopefully use to exploit. So based on this info that we have, what are our options for developing treatments? So there are kind of 4 categories that we're going to go through, 4 different types of treatments, or 4 different options that we could explore. So the first thing is we could just invent a new drug. So I have it down here as create a new small molecule drug. So what this means, this is kind of arbitrary terminology, but effectively what a small molecule drug is something that is not a biological molecule. So we're not talking about a gigantic protein that has many, many thousands of atoms and a very complex structure. We're talking about something more like Tylenol or aspirin. These are small molecule drugs. They have, maybe kind of 25 to 100 atoms in the molecule. Again, much, much smaller than these very large biological molecules. When I worked in pharmaceutical research and development, I worked on small molecule drug development. So again, it's 2020. We've been able to completely characterize this virus in less than 6 months. How hard could it be to develop a new drug? So why would we want to do a specifically small molecule drug in the face of the other options that we're going to go through shortly? 2 of the big advantages of these so-called small molecule drugs is that most of the time they can be given as a pill rather than as an IV injection. So again, things like, think aspirin, Tylenol, ibuprofen, things like that. The second major advantage of these is that it's relatively straightforward to mass produce them, much more so than some of the other options. The problem is that creating a new small molecule drug is not at all easy. So research and development, and specifically pharmaceutical research and development, is often compared to setting a gigantic pile of money on fire. This pile is nowhere near large enough, even with the U.S.-Canada exchange rate at what it is at the moment. People have done studies on how much money does it actually take to create a new drug, and estimates vary wildly. It really depends on a huge number of factors. But just for our purposes here, just so that we can get a sense, a pretty good estimate is about $3 billion. That's a lot of money. But in the current circumstances, we're losing tons and tons of person hours of productivity. The economy is in the tank. $3 billion sounds like a bargain. This is not really going to be that much of a hurdle. A second problem, though, is that even with a lot of money being spent, the odds of success are actually pretty low. Despite all of our knowledge about biology and pharmacology and how drugs work, the drugs that we've developed, how do they work, and with a lot of effort behind it, it's still a very risky prospect, and most molecules, most candidate drugs do not make it through to full development. Again, the estimates here are have a huge range, and it's kind of difficult to actually even define what you mean by rates of success. But one kind of measure, again, just to give us a sense, is about 1 in 10,000. So this would be for every new drug that we want to get across the finish line, we're going to need to make about 10,000 molecules and test them to find that one drug. This is the mother of all needles in haystacks. Now, again, making new molecules is not easy. This is what my group and I do for our livings. But again, in the current circumstances, it seems like we can get all hands on deck and just put as much effort behind this as humanly possible, and crank through as many candidates as we can and increase our chances of getting something across the finish line. This is totally doable as well. We can put a lot of eggs in a lot of baskets and see what we come up with. But there's still one other huge problem. And unfortunately, we're not going to be able to get around this one even in the other modes of, or the other options that we're going to talk about, and that's time. It takes a long time to create, invent, and study and get approved a new medicine. As with the other factors here, again, the timelines and estimates vary a lot, but just for our purposes, a pretty good estimate is about 12 years. Now, this is obviously not going to help to impact our current situation. 12 years here sounds a little bit extreme. Why does this actually take so long? Making 10,000 molecules, for example, certainly does not take 12 years. Really, the reason that this takes such a long time comes back to Feynman's quote about not fooling ourselves. We need to make absolutely sure that any new medicine that we create is both safe and effective. It's not much good to us if it's only one of those things. And so what this means is that we need to do sophisticated clinical trials that last months and sometimes years. This is where most of the money in our burning money pile is invested into. Identifying potential drug candidates is not nearly as difficult or as fraught with failure as actually getting them through clinical trials. So the gold standard for a clinical trial is what's called placebo-controlled. It means that some patients receive the drug and others receive some inert placebo to account for the bizarre placebo effect, where people will spontaneously get better even though they're taking a sugar pill as opposed to the actual drug. And these trials also need to be what are called randomized. And so if you have multiple treatment groups, maybe you're looking at different doses. Obviously, you're going to include this placebo-controlled group the enrollment of patients in those groups needs to be random. We need to account for any kind of hidden features, hidden factors that might influence how the influence the outcome of this clinical study. So each of these patient groups needs to be as similar to one another as possible with totally random participation. These also need to be what are called double-blinded, in which case not only do the patients not know whether they're getting the drug or the placebo, but the people who are the doctors administering them don't actually know either. And this is done to prevent any biases from creeping in. All of this, again, is to make sure that we don't fool ourselves into thinking that we have something that is working when actually it's not. And then we wind up wasting a lot of time, effort, and money chasing down something that is not that's not real. For a lot of these reasons, most potential drugs that enter clinical trials end up failing. For infectious disease, the failure rate is not as bad as in some other disease indications. So it's about 85% success rate. So 3/4 of potential anti-infectives fail during these clinical trials. And so this is really what's limiting us from going any faster. There's certainly a lot of value in starting from scratch like this and developing new small molecule drugs to treat COVID-19 and coronaviruses writ large, but it's not going to come in time to impact our current pandemic. The herd immunity will have set in one way or the other before we get any of these out of the gate, basically. So, given that this is not really an option for the near term, what else can we do? So we could repurpose an existing drug. So rather than going to the pains of developing something from scratch, we could take something off the shelf and see if it'll work. The FDA has approved about 1,700 drugs in its history, so we have quite a few options. And the really great thing about repurposing a drug is that we already know something about it. We know how to make it. We should know a lot about its safety profile, and we might even have an idea about how it might actually work. So, for example, a drug that has been developed to treat one type of virus may function in another type of virus through a similar kind of path. So, repurposing drugs from one disease to another is actually pretty common. And before we get to the COVID-19 examples, and there's a couple of pretty high-profile ones, one of which I'm sure everybody is well aware of already. I'd just like to go through an historical example of drug repurposing that kind of encapsulates a lot of these things that I've talked about so far. And likely many members of the audience will be familiar with at least part of this story, and that's the story of the drug thalidomide. So thalidomide is probably one of the most infamous medicines that's ever been developed. It was developed in Germany back in the mid-1950s. It was available over the counter and was given to advertise for things like trouble sleeping, anxiety, and also given to pregnant women to combat morning sickness. Now, very long and very painful story short, this drug causes birth defects, and this wasn't fully recognized in some of these places until after a sizable number of children had either died or been burdened with very debilitating birth defects. By 1962, it had been pulled from all markets and banned, including Canada, where it was also a big problem. But one place that never actually saw widespread use of thalidomide was in the United States. And this was due to the heroic efforts of an FDA case manager, Francis Oldham Kelsey, who refused to approve thalidomide because there was not sufficient clinical safety data. So despite multiple attempts by the company to get this thing approved by in the US at the FDA, Frances was not, she was not fooled. She did not approve this, and as a result, I believe there were only 17 examples of, during investigational studies, 17 cases of birth defects in the U.S. Now, something really interesting about Frances that I had no idea about until I was researching for this talk. She was actually born and raised right here on Vancouver Island in Cobble Hill, up in the Cowichan Valley. She attended St. Margaret's School and also attended Victoria College, which was the progenitor school to UVic. So she is actually technically a UVic alumnus. Now, this is the part of the story that most people know about thalidomide, but the story's not over then. So thalidomide was actually repurposed shortly after this as a treatment for leprosy. So this was discovered, somewhat by accident by a doctor in Israel who was treating leprosy patients who were, having a really, really hard time sleeping, and nothing was really working. And so this doctor knew about thalidomide. He knew the risks, but in the face of the unmet need, decided to try thalidomide to help his patients get some rest. Not only did it help them sleep, but he actually saw the condition improve. So this is very, very unexpected, and led to kind of a resurgence of thalidomide as a treatment for leprosy. Even though this was discovered back in 1964, the FDA didn't approve it until 1998, more than 30 years later. It is still used today to treat leprosy, although there are other treatments that are even more effective and don't have the same kind of safety problems that thalidomide does. But the story is not even over at this point. So around the time that the FDA was investigating whether thalidomide should be approved for leprosy, it was discovered that it actually is a highly effective treatment for blood cancers, specifically multiple myeloma, as well as a few others. So this fact was discovered in 1994, and in 2006, thalidomide was given accelerated approval by the FDA to treat multiple myeloma. And one, kind of close molecular derivative of thalidomide is actually one of the most prescribed drugs in the world, for these blood cancers. So this is a very long 50-year history from thalidomide's initial discovery through to what now has become its kind of main medicinal use through this process of drug repurposing. So what are our options for COVID-19? There are quite a few known drugs that are being investigated. Again, they're undergoing clinical trials right now to make sure that, again, we're not fooling ourselves. But I'm just going to talk here today about two that are kind of illustrative of the challenges of repurposing an existing drug for something like an infectious disease. So the first one, which I am going to say a lot less about than I had initially planned, is hydroxychloroquine. So everybody has seen this in the news. This is a very old drug. It's about thalidomide vintage. It was FDA approved in 1955 to treat malaria. It's used less for that now, just due to things like pathogenic resistance, but it's used extensively in some autoimmune diseases, things like lupus and rheumatoid arthritis. And so again, it's a very old drug. We know a lot about it. This was initially looked at as a potential treatment for COVID-19. This is the timeline, and again, I don't really want to get into the whole thing here. The politics of it is something that I want to stay kind of completely out of. But just to sort of point out that this whole thing with hydroxychloroquine started with a preliminary anecdotal report on its use in France by this guy, Dr. Raoult. This was 24 patients. There was no randomization, no proper control group, certainly not anywhere even close to this gold standard clinical trial that I mentioned earlier. But there was a hint of possible benefit. Something that we definitely needed, just as a species, as a civilization, back in March. Now, the intervening time has seen a huge amount of work done, studies done, none of them really approaching that same level of rigor that we need in order to be sure that we're not fooling ourselves. Some of these papers have been retracted because the data they were based on was not sound. Again, this is just a whole can of worms that I'm going to leave mostly closed here. Very recently, so last Friday, a large and high-powered trial called the Recovery trial out of the UK, this is again one of these gold standard trials, released some information that they saw no benefit when they conducted this carefully controlled study. Now, there's a lot of other trials that are still underway, things like combination therapies of hydroxychloroquine with other drugs. But right now, the best information that we have is that this is not effective. And so we probably should move on to something else at this point. So a second drug that is showing a little bit more promise is remdesivir. So this is not actually currently approved for anything, but it's been studied for a number of different to treat a number of different viral infections. And so that vir at the end of the name means that it's an antiviral. Unfortunately, it's not orally active. So this is an example of one of these small molecules that's we still actually do need to administer by IV, which dramatically sort of reduces its attractiveness. However, we do know that it will interfere with the virus's RNA duplication. So again, this is one of these proteins that I talked about that is critical to the virus's function. We've actually, there have been studies done that show that remdesivir is able to get in there and throw a wrench into that replication process. So we got a little more, kind of confidence that this repurposing effort might be worthwhile. So this compound was originally developed to treat hepatitis C, but unfortunately it didn't work. In the aftermath of that, it was actually tested for quite a while against Ebola, part of a number of studies funded by the U.S. Defense Department, and it was deployed during the Ebola outbreaks throughout 2013 to 2019. It did show some signs of efficacy, but ultimately there were other treatments that were better. So its use for Ebola was kind of marginal. The nice thing about all of this prior data is that, again, we have a pretty good idea of how to use this drug, what kind of doses are going to be suitable, the safety profile, things like that. And so a few weeks after hydroxychloroquine kind of burst on the scene and sucked up all the oxygen in the discussion about repurposing drugs, there was a compassionate use study that was reported. Again, not controlled, low numbers of patients, but again, something like the initial hydroxychloroquine work that showed some promise. And so this was followed up by very, very quickly by a much more rigorous study that was controlled, this so-called ACTT1 study in 27th of April, so a few weeks later. And this did show, again, some benefits, an improved recovery time in patients who were not, did not have severe disease. The data as we have it right now is still not really conclusive. It does seem like there might be some benefit, but we don't yet have that gold standard, large controlled study. Even if remdesivir does turn out to be effective, it's not going to be a cure. It's not going to be, the thing that turns the tide here. It may offer some benefit for certain patients, but it's definitely not going to be the main thing. So where does that leave us? The next option we have is that we know we need to gum up the works with respect to the virus proteins. Why don't we fight proteins with other proteins? So this is the approach that our immune system uses. And so we can take a lot of inspiration from our immune system and use what are called antibody-based therapies. So an antibody is a protein that gloms onto another protein. It grabs on, very specifically and very, very tightly. So this can do things like tag viruses or bacteria for removal by the immune system, and it can also do something like cap that spike protein. And so you've probably heard a little bit about antibodies in the context of testing, antibody-based testing to see if you've had the disease. So when we have an infection, our body raises antibodies to fight the disease off. In addition to testing, we can actually use these antibodies that people generate during infection as treatment. So this is actually a really old technique. It's called convalescent plasma treatment or serum treatment. This was actually one of the things that was used back in 1918, 1919 to treat the influenza pandemic. So basically what we're doing here is we take donated blood plasma from someone who survived the disease and use the antibodies that are present in that from their immune system to kind of kickstart the immune system of somebody who hasn't yet had a chance to mount that response. The upside to this is that it's something we can do really quickly. This is happening now. It's been happening for quite a while. The downside is that, well, there's two downsides, two big downsides. The first is that there's a high potential for variability. So, you're basically relying on the immune system of the donor to have mounted a strong response in order to get a lot of these antibodies in there. And every donor is going to be unique. Related to that is the second problem, which is very hard to scale something like this up. Each donor can really only provide plasma for about three patients, and so trying to scale that up to the level where it could be used, in a large patient population is very, very, very difficult. But again, it is something that we can do and are doing right now. But we still need to determine again if this is actually going to work in a clinical setting. So far, the results are not clear. There was a recent study that's read out last week looking at convalescent plasma that again seemed to show some benefit with respect to recovery time. But unfortunately, the enrollment in this study actually was halted before they reached their intended target, and so the data that came out of it is not definitive. And so this is kind of a recurring theme of these. We're still at the point where we're waiting for these clinical data to read out. So plasma is, we're relying on the immune systems of donors to have raised these antibodies to combat the disease. Can we do better than this, though? And so one, much newer technique is to identify, isolate, and manufacture specific antibodies that are going to be targeted to a specific protein for the virus. And so these are what are called monoclonal antibody therapies. This is some of the most impactful and most prescribed drugs in the last 10 or 15 years are antibody-based therapies. They're used for a lot of different indications. In the current case, this is probably our best bet in the short term to create something that is going to be impactful. And so this image here just shows again this spike protein kind of coming off the surface of the virus in kind of off-white orange, bound to an antibody in pink and red that is basically capping that spike and stopping it from being able to infect another cell. And so monoclonal antibody therapies, there's a lot of work that's going on here, and we're just starting to dose the first examples in human patients now. So last week, I believe, was the first clinical trial with a monoclonal antibody therapy for COVID-19. It started, and it's actually using an antibody that was invented by a company called AbCellera that's based in Vancouver. And so we'll obviously need to wait for the results to come out for that, but again, this is something that, in contrast to that very long lead time for a small molecule drug, an antibody therapy is something that we can get off the ground fairly quickly. So the last thing in the couple of minutes I have left here is to create a vaccine. This is the big one. This is what we're really all waiting for here. And so vaccines give us, they're very closely related to antibody therapies, in that they're a way for our immune systems to actually get a sneak preview of a disease or a virus and be able to raise the antibodies that are going to combat it and stick to those proteins and stop the infection before we actually get infected. So this has been a hugely impactful thing in modern medicine and in public health. Again, this is responsible for things like eradicating smallpox and basically eradicating polio. To what miraculous being do we owe vaccines just as a concept? And it turns out it's cows. The word vaccine actually comes from the Latin word for cow. And this is because the first vaccine that was ever created or identified was the cowpox virus that was used to inoculate people against smallpox. And so the smallpox vaccine is still sort of based on this technology. This was invented back in the late 1700s. Unfortunately, for as long as there have been vaccines, there have also been anti-vaccination movements. So this is just a caricature of, published by something called the Anti-Vaccine Society in 1802, claiming some pretty interesting side effects from inoculation with cowpox. You can see a lot of people developing cow-like growths all over themselves. And so the more things change, unfortunately, the more they stay the same, it seems. So with respect to vaccines development, the U.S. government has framed its efforts as Operation Warp Speed. Have we reached Warp 9 yet here? And so, as with the other examples I've talked about, we're making good progress, but it's there's still a lot of work to do. I'm just going to briefly talk about 3 different varieties of vaccine, and all of which are being investigated for COVID-19. So the first major class are these so-called inactivated live viruses, or attenuated live viruses, or inactivated viruses. So these are, examples of these are the MMR vaccine, or the annual flu shot. Basically, here we take a weakened or dead inactive virus and just give it to the patient, and let the immune system kind of pick over the carcass of the virus and get an idea of the what proteins there are, and able to raise the antibodies to combat them. These are being developed. Vaccines of this type are being developed. There's a couple of companies in China that are starting to enter early clinical trials, but we don't have any data yet on whether or not viruses or vaccines of this type are going to be effective. A second major class of vaccine actually only delivers kind of parts of the virus. So these are kind of the key proteins or other key bits that the immune system can recognize and again create these antibodies to bind to them. And so things like the hepatitis B vaccine and the shingles vaccine use this kind of approach. So the advantage here being you don't have to give a potentially infectious agent to your patient to be able to get this immunity. Again, there are a lot of companies and universities that are looking at this particular mode, and basically what they're looking to do is deliver that spike protein, on its own as the key piece that we need the immune system to recognize. But again, we're still waiting for data. The final class, and so this is a really large class, and I don't have time to go into all of the intricate details here, but basically a huge number of programs for COVID-19 specifically are using a different approach where they only give the blueprints for proteins. So rather than delivering this spike protein in a vaccine, they're actually just giving DNA or RNA blueprints that would tell our cells to create that. And so this is probably at least the majority of programs that are far along at the moment. All of Operation Warp Speed's programs fit into this category. The downside, as you'll notice here, is that I haven't given any examples of this in terms of vaccines that we're familiar with. To my knowledge, there's only one approved vaccine that uses this kind of technology, which is an Ebola vaccine. It was actually developed in Canada in the early 2000s that received FDA approval in December of last year. So this is really, really new technology. It's being explored really heavily because it's quite fast to develop. Attenuated live viruses or inactivated virus types are a little bit more work. And so this is kind of a way to get some potential candidates very quickly, but unfortunately it's almost unproven technology. And so we need to wait and see what the clinical trial results say. So unfortunately, it's kind of ending on a bit of a maybe not quite a downer, but we still have a little bit of ways to go with respect to this. So I just wanted to very briefly talk about probably the most effective thing that we can do and are doing against COVID-19, which are non-pharmaceutical interventions. And to end on an optimistic note, certainly in British Columbia, we have really crushed the disease curve and are in a really good spot with respect to controlling the disease. And so we are going to need to keep doing this sort of thing for a little bit longer while we give these clinical trials time to give us confidence that these potential new treatments are actually going to work. So with that, thank you. I apologize for going a little bit over my allotted time, but I'm happy to answer any questions that anybody has.
Well, that was a wonderful talk, Dave. I'm sure everybody watching will want to join in a quick virtual round of applause. So if you're watching us, please click on your raise hand as a way of showing your appreciation for Dave's presentation this evening. I certainly learned a great deal. You can pop it down once you've raised it as well. We like the enthusiasm. What I would also say is, for those of you who want to ask a question, let me navigate you through that quickly. Just hit the little magenta tab with the two arrows on it at the bottom right of your screen, and you'll open up a little side window. If you click on the little speech bubble at the bottom that says chat, that will give you access to a little bar where you can type in your question. So we'll just have a quick back and forth, Dave and I, while you actually type any questions that you may have. I guess one thing I wanted to draw attention to, Dave, is to give you a little bit of additional kudos that some of our participants may not be aware of, and that's that during the last couple of months, you were quite instrumental at UVic in using the spare supplies in the chemistry department to make 200 liters of hand sanitizer and donate it to Island Health.
Yeah, no, thank you for recognizing that. Yeah, we had a lot of ethanol on hand, and it wasn't really doing much good sitting on the shelves. And yeah, there's a really great WHO protocol for kind of field manufacture of sanitizer. And so yeah, we whipped up a few batches and managed to donate a pretty sizable amount to Island Health. So it was
Yeah. Now, as I said, feel free to type in your questions into the chat bar if you have a question for Dave. I guess one thing I could ask while we're waiting to see if there's any questions, Dave, is not if you're let's not call you a betting man, but let's say you're a stock investing man. Where would you be putting your money right now in terms of these cures?
That's a really good question. So I think in terms of speed, something that we can get that's going to be actually effective relatively quickly. the monoclonal antibody therapies are probably the things to watch for. So again, they're a little bit behind in terms of getting into clinical trials. You know, these drugs that have been repurposed and some other things have already had some trials done. But just because of the specificity and the fact that again we know that this is able to glom on and shut down that spike protein, that's probably going to be the best sort of near term thing. In terms of the vaccine, you know, which type of vaccine is gonna work, I don't know. There are no coronavirus vaccines yet, so that's an open question.
Yeah. Well, we have a couple of questions now, so let me just read them out one at a time in the order they come in. So the first is from Dave McMurtry. Do RNA vaccines work by producing viral proteins that then provoke an antibody response?
Yes, yeah, that's exactly right. Yeah. So there are RNA vaccines and DNA vaccines, and the idea between both of these is that there are and then there are a lot of different ways to get these things kind of into your into your cells. But the idea there is that you use like the virus does, you use the cell's own machinery to produce the proteins. One of the advantages to this is that because the cell is producing the proteins in the way that it would, with a viral infection, they come out kind of folded properly and in the right form, the same kind of analogous way that they would in an actual infection. Um, the downside is that they're still unproven, and so remains to be seen if a strong enough immune response is going to be raised to them.
Okay. So our next question is from Dan, and his question is, how long are the clinical trials going to run for, and have all the countries grouped together in relation to an approval process?
Yeah, no, this is a really good question. The trial length, I mean, as you saw from some of the examples here, some of these trials are already starting to read out. Um, it really depends on, so that an individual trial, the length of time depends a lot on how many patients you're looking at and kind of what mode of treatment you're trying to investigate. So for these, for something like the remdesivir trials or the hydroxychloroquine trials, again, we've already got enough stocks of these things available so they can be distributed to to the clinic sites for use. The numbers of patients have so far been, not enormous, and so getting the enrollment, which is something that can take a lot of time, um, is a little bit easier. And the time to actually see a response is relatively quick. I mean, we're looking at times to recovery in the sort of 10 to 14-day ballpark. So to get any individual data point, it's actually relatively fast. What makes those kinds of trials take longer is this enrollment question. If you want to have a very large and very highly statistically powered trial with enough people in it, you need to find all of those people, and you need to make sure that again, your inclusion criteria, so that the groups are all well matched and things like this, that you've got the right patient population. So that can often take quite a while. Um, with respect to the vaccine trials, it's a bit of a different question, because there what you're looking for is protection against infection. And so unless we're willing to do the sort of ethically nebulous route of actually trying to infect people, so giving them the vaccine and then challenging them with the virus, um, giving a vaccine to something like, frontline healthcare workers and then waiting to see who develops it, that's going to take a lot longer. And so the vaccines development from a clinical trial perspective is going to be, much more in the, many months sort of time frame. With respect to the approval process, that's a really good question. And I'm not sure exactly how this is going to go down, but we're not quite there yet, just with respect to where these treatments are. But I don't imagine that there's going to be a whole lot of daylight between, the European Medicines Agency, the FDA, Health Canada, things like this. Yeah, that remains to be seen if there's going to be coordinated effort, or if it's kind of business as usual, and the documents need to go out to all places.
Well, I guess we have time just left for one quick question. I don't ever like to cut people off when they've gone to the trouble of typing it into the chat bar. So this is from Gayla. What's the difference between the vaccine development process for, say, COVID and for something like HIV?
Oh, that's a really good question. So again, I'm not an expert in vaccines development. Um, so HIV is one of these these things that we've known about it for quite a while, and yet no vaccine has been developed for it. Um, I don't know enough about the actual virus to comment on that. Um, so yeah, I'm sorry if that's not a very satisfying answer, but
No, no, that's fine. You've done a great job this evening. You've gone above and beyond. What I might do now is I'm not going to have time for any more questions because I don't want to keep our audience too long from whatever they've got planned this evening. Let me just, before we close up this evening, give our audience a very quick advertisement for next week's talk. That's going to be same time, same place, same link. That's going to be another UVic chemistry professor, Catherine Elvira, and she will be talking about modeling the gut on a chip for drug discovery. So keeping up a bit of a theme in our talks in June. Um, don't worry if you haven't got the link. We'll be emailing that out to you all tomorrow with an advertisement. And don't forget, if you want to recommend this talk to one of your friends or colleagues, it will be appearing on YouTube as well, UVic Café Sci, in the next couple of days. And so there's nothing more for me to do this evening apart from invite you all to thank Dave once again. If you want to give one last raise of the hands, and then you guys are all free. You've had a great time this evening. There we go.
thank you for all
Raise
of
hands
the
across the
the
floor.
dings.
Yeah. Well, thank you very much, Dave. That was a wonderful presentation, and I think everybody's had a great evening. So thank you very much.
Yeah, thank you again for inviting me, and I look forward to the next one.